Hello to everyone, I am Professor Masood Fuzail. In this video lecture, we are going to discuss some important and early stages of development of frog or development in amphibians. So let's start this lecture. So I am going to start with fertilization. As you know that when breeding season start, female frog lays millions of eggs, and at the same time male frog sprinkle sperms on the surface of eggs where eggs are fertilized. If we look at the structure of an egg, this egg of a frog is much larger and it contains two poles which are known as animal pole and the vegetal pole. The animal pole is on the top of the egg, which is little bit darker and contain nucleus, while the vegetal pole is lighter in color. There is also a presence of grey crescent. Grey crescent is a specialized area on the surface of egg which appears after fertilization, right across the surface where sperm has entered in the the sperm has penetrated in the egg. Eggs are laid in the metaphase plate and in the metaphase stage, and after fertilization, the development process of an egg starts. And the first step after fertilization is of the development of the frog is cleavage. Cleavage is a simple cell division with help of mitosis in which a single zygote divides into many cells. First cleavage usually completes after 3.5 hours of the fertilization. In this condition, single cell is divided into two equal amount of cells. Yolk, cytoplasm and nucleus has been divided into equally with both cells. After 4.5 hours, second cleavage has been completed and the embryo has been divided into 4 cells. And after 5.4 hours, third cleavage completes and embryo has been divided into 8 cells. And this process continues, and after 7 hours, the embryo has been divided into 32 cells. After this, it will become 64 cells and then 128 cells, and this embryo will be converted into morula, which is a group of cells which are known as blastomeres. Morula stage is short-lived, and soon this morula changes into another form of the embryo, which is known as blastula. Blastula is a group of cells, hollow ball of the cells, which contain a cavity which is known as blastocele. So blastocele is a cavity which is the sign of the stage which is known as blastula. Blastula contain blastocele and also contain yolk, and the cells of the blastula keep dividing, and now this blastula is going to enter into a next stage which is known as gastrula, which is very important stage of the development. During gastrulation, germinal layers are formed, and from these germinal layers, which are ectoderm, endoderm and mesoderms, organogenesis will take place and different body organs of the embryo will form. So let's discuss how this takes place. If you look at this diagram, so the blastula, the cells on the outer surface of the body, start pushing inward inside the blastocele. As you can see right over here, the cells of the outer surface of the blastula are pushing inward, and some of these cells, which as you can see in the green color, these are presumptive notochord, because these cells are going to form notochord in the future, while the blue color cells, as you can see in the diagram, they are going to develop neural plate, which is going to form nervous system for the embryo. So the cells are keep dividing and start pushing towards inside, and the blastocele cavity is reducing in the size. Now let's move on to the next diagram. In this diagram, as you can see, this is the outermost layer of the blastula. This is the outermost layer of the blastula, and the cells from this side, when pushed inside, which is known as emboly, they move inside, and the blastocele has totally has been disappeared, and the cells of presumptive notochord and neural plate are joined with the ectoderm or epiblast, and there is a cavity develops right over here which is known as archenteron. This is going to form gut of the embryo. The dorsal portion of this cavity is known as dorsal lip, and the lower on the lower side there is on the ventral side of this cavity there is a lip which is known as ventral lip, and the cavity, the opening of the cavity is known as dorsal pore. So now let's move on to the next diagram, which is a little bit more clear. The outer layer is known as ectoderm of the cells, and the inner layer which is known as endoderm. So the both of the layer has been formed, ectoderm and mesoderm, and the cavity between these two layer is known as archenteron, and the opening which is remaining right over here is known as blastopore, and the dorsal side is known as dorsal lip of the blastopore, and the ventral side is known as ventral lip of the blastopore, which are very important role in the formation of the nervous system. And this pushing of the cells is known as, which is outer layer of the cell into the cavity is known as emboly, and the two layers which are present up and down is known as epiboly. So now let's move on to the next diagram. So in this diagram you can see that this is the ectoderm of the embryo, and this is the blue color in the diagram is the endoderm of the embryo. Now there at the same time ectoderm cell divides and start to form mesoderm. So as you can see in this diagram, the red color cells are mesoderm, and blue color cells are for neural plate, and the green color cells are for notochord formation. Now if you look at the next diagram, the ectoderm has been completed and completely surrounded the yolk. Endoderm is also forming, mesoderm is also developing, neural plate cells are also dividing, and at the same time future notochord cells start dividing, and this is the cavity between these cells which is known as archenteron. As I mentioned earlier, this is going to be developed into the gut. So this process of the gastrulation is very important, because in this process germinal layers are formed which are known as ectoderm, mesoderm and endoderm. These germinal layers are going to form different organs in the body, a process known as organogenesis. For example, ectoderm will develop skin, brain, spinal cord, receptors, neurons, etc. Mesoderm will going to develop notochord, muscle cells, blood, bones, sex organs, while the endoderm layer will going to develop gut, lungs, gland, liver, pancreas, thymus and etc. So now let's move on to the next diagram. In this diagram you can see that the ectoderm has been completely surrounded the embryo, and endoderm is going to form gut, and neural plate is also going to develop into nervous system, and the notochordal cell are going to form notochord, mesoderm also dividing to form a coelom. So all the cells which have been differentiated and going to perform their function accordingly. In the last diagram, as you can see, there is a ectoderm has been surrounded the whole embryo. Endoderm has been developed into a structure which is known as going to be digestive system, and the cavity between them is a gut. And notochordal cells has been developing notochord, and you can see in this stage a notochord is developing inside the embryo. Mesoderm cells have been spread all around the embryo, and they are going to develop their internal organs of the body according to the rule. And the neural tube cells are going to form nervous system. So there is the cavity between mesodermal cell which is known as a coelom. Coelom has been developed right over here. Neural tube has been developed, neural tube, and at this stage is called neurula. Brain, spinal cord and brain are also going to form from these structures. When all these germinal layers has been formed, then the next process of the development starts, which is known as organogenesis. And these different layers and different parts of the embryo are going to develop into different organs of the body. Soon this embryo will develop into a larva which is known as tadpole larva, which has tail, gills, legs, and start feeding on the small planktonic creature present in the water. and then later in the process known as metamorphosis, this tadpole larva changes into an frog, a small tiny frog, which later on develop into an adult frog, and the life cycle of the frog is completed. So in this lecture I try to give a concept about early developmental stages of the amphibians and frog, and I hope it makes sense. And that's all for today, and I'll see you in the next lecture. Until then, bye.